All right. Let's start. Hello. Good morning. Good evening. We have folks across different time zones today. Before I begin, I wanted to thank you for tuning in today, where I'll be sharing my team's experience and mine so far with the G4X instrument. As a brief intro, I'm an associate professor at Harvard Medical School, and my lab is located at Beth Israel Deaconess Medical Center in Longwood in Boston. As a faculty, I'm also a member of the Dana-Farber Harvard Cancer Center and the Broad Institute of MIT and Harvard. Apart from the lab, I'm directing the Spatial Technologies Unit, which is an institutional BIDMC core, and now it's also a Dana-Farber Harvard Cancer Center core facility. The lab and the core are two separate entities. They have different missions, personnel, budget, space, and worldviews. And I'll try to present today my thoughts a little bit as a PI of a research lab that uses spatial omics and focuses on spatial biology and especially translational research, but majorly as a director of a spatial tissue profiling core facility. Um, let Okay, changed. So before I begin, some important disclosures. I will be discussing openly today what we have seen so far. We have the instrument, multiple instruments actually since December 2024, and I have received the okay to mention everything, as well as what we will be testing in the next couple of months. I think that's very interesting as well. Even though this webinar is organized by Singular, uh, that has nothing to do with my opinions. They're all shared today, are my own and my team's. And no, I haven't been paid for this webinar in any shape or form, and I'm not consulting for Singular. So I usually don't have to be that specific, but I think it's better now than at 3 a.m. on Twitter or WhatsApp. So on the top, you can see my disclosures. Well, the facility, which is on the bottom, is performing often early access programs with selected technology developers. We take every early access very, very, very seriously, and which I will be discussing our strategy a bit. And always, we're happy to share our experience after it's over or when it hits an interesting milestone, such as today. So let's talk about the lab a little bit. Our lab research focuses a lot on self-known self-recognition, how our immune system is able to distinguish who is friend and who is a foe. And our research projects usually span from autoimmunity all the way to cancer immunology. I would say one third autoimmunity and two-thirds towards cancer. And most of our work follows the reverse translation pathway. So we learn from the patient and we go back to ex vivo, organ on chip, in vitro, in vivo, and then back to the patient. And in order to do that, we try to learn as much as possible from patient samples and extract the last ounce of information. So we use single cell, we use a lot of different technologies, and for the past 5 years, I would say a lot of spatial, which I will be discussing today. And these are I would say, five major research domains. Translationally, we work in the lab. We have different projects, and some are more mature, some are in early discovery, others are in clinical trial correlative phase, others are in phase one or two clinical trials. So there are a lot of things happening in the lab. And to support that research program, we focus a lot on tissue. We pay a lot, on patient samples, tissue architecture, and most of our work nowadays is in situ. And the tools we have today they're incredible. There's I would say since 2020, maybe 2019, the options we have to interrogate tissue are quite phenomenal. And it's like having the most powerful microscope together with the sharpest scalpel. And that's the dream for generations of physicians. But as you see, it's not a new thing. I'll discuss that in a bit as well. And to support these investigations of our group and others, there are a lot of different technologies, being released for the past, I would say 4 or 5 years, majorly, and we often see them in these like two-dimensional maps, like this one in papers and reviews, and majorly is a distinction between plexity and resolution. And that's like the PI, my major PI mindset thinks about this when we're selecting things. But um, sorry, but as a you know as a provider of these spatial tissue profiling services, um, that's not 100% what we think. And the more you use them as a PI, you start thinking alike. So the robustness of the assay, are you going to lose samples or not? That you care about as a core facility a lot, but also as a PI as well. The ease of use, the ease of customization. Can I create my own, panels? Can I create my own like customized experiments? The imaging real estate, how big of a sample can I interrogate or how many? The throughput of the assay, how many per month, per year? If I want the project to be done that has a clinical trial and I have 10 samples, 50 samples, 100 samples, how much as a PI will take me? But if I'm a facility, how many projects and how many PIs I will be able to help, within a year. Its robustness as an instrument, standardized best practices, how is the data analysis? Is it mature or do I have to reinvent the wheel? Does it support any type of sample from different organisms, FFPE? All matter, as well. So I think, we might have different weights when, depending on the research questions and depending whether you're a PI or a facility. But I think all of them, the more you use spatial tissue profiling, the more you think about them. So now that I introduced a little bit the lab and the mindset of the lab, I would like to spend a few minutes to introduce the facility. And because the facility is hosting the instruments, and that's where most of the work has taken place, that's the major hat I will be wearing today. Um, so the Special Technologies Unit is started as a new venture back in 2020, and we wanted to establish a freestanding center focused exclusively on single-cell spatial. And we opened doors to the community in 2021, and that was enabled, it sounds like very close. Uh, but spatial years in this field are like dog years. Every year there are advances that happen, the equivalent of seven normal years. And we had a lot of support from the Commonwealth of Massachusetts, BIDMC, the Harvard Medical School Initiative, for RNA Medicine. Lots of people helped to launch this, and it's a center focused till 2025 September exclusively on spatial and single cell technologies. Some things that make it unique, I think, is an outwards facing facility supporting not just the hosting institution, but also, academia, industry. And it recently became a core facility of the Dana-Farber Harvard Cancer Center, which is a broader community. Um, it When it was established back in 2020, it was done so through a quadruple mandate. Number one, of course, to provide all our, equimolar, all have the same focus and attention. One is to provide democratized access to technologies to everyone, regardless whether from academia or industry, not just the labs that are closest to the technology, or the research labs that are deeply embedded already in spatial. It supports equal investigators who have the extensive expertise in the field, but also those that have, they're, for instance, in a clinical environment and they have tissue and a question. And I think a lot of the innovation and the findings come from both groups. Training and education, which is the second pillar, is not just for the researchers who do the experiments or they want to do such experiments, but also to PIs, physicians that want to leverage such techniques for their study or grant design, but also students, investors, decision makers who want to better navigate the field that is changing so rapidly. And then, of course, support the generation of new IP, innovation, entrepreneurship, support of the creation, new assays, new products, spin-offs, spin-outs, startup, and different types of industry collaborations, which practically this early access is part of that mandate. And as well as pave the way for spatial tissue profiling to reach clinical care and benefit patients. So in terms now of access to technologies, very briefly, the facility serves the community by providing, access to different assays across the resolution axis, across the throughput axis, and they have all been selected. It's not a collection of instrumentation. They have all been selected based on supporting very, very specific and distinct needs. And there is a very lengthy process of how we select what we bring on board. And the facility is a complete and autonomous ecosystem. Our team, processes the tissue. We can do tissue microarrays to increase throughput and reduce cost, of course, with the penalty of the size of the tissue. We have a lot capabilities for doing single cell from the same blocks, from the same sections. Extensive robotics and automation capabilities in order again to have increased throughput, reduce costs and labor. We do our own sequencing in-house, the data processing in-house, and the data analysis. So, and by internalizing everything, of course, it's a lot of work and it's a massive investment. But on the other hand, you control your timeline, you control the quality. There are benefits, through that, especially if you're supporting clinical trials, if you're supporting large consortia. That control, I think, is very important. And I mentioned like very briefly the IP support and generation, and that is also, early access and supporting a company who's building a new assay is a part of that, and we take this very, very, very seriously. So it's a conscious decision. It's not let's test an assay that's for us and see if we want to use it or not. This is something that's part of our mandate, and we do this year round. And also it's important for people who don't work in a core to know that it's not the cheap thing to do. So even if you, let's say, but you get the free kit, you have to put people, somebody will have to perform the experiment, analyze. Even if you ship the samples out, there will be, a computational biologist who will spend time to report, benchmark, assess. Then teams will meet. So you're removing that person from production for that time. So usually, we internalize the assays. We try to see them on the bench. So that takes, wet lab personnel for the people who will be doing the experiments, combio, administrative, project management, for instance, leadership meetings, space, if it's an instrument, networking, computational capabilities, data space. There is, depending on the type of early access, it's a small, medium, or large investment. So we cannot do it, 50 times a year. There is a specific threshold, and we take into account different things. For instance, is how big is the community need? Is it something that people have asked, to do, or we think, there will be it will make a big difference. It will have an impact. There will be a return of investment, down the road. What is the timeline, for instance? Is this something super mature and they don't actually need us? Or is this something that, it's a two-way communication street and we can potentially help and shape it in a way that we think will serve, the community better. So, and how do you assess impact? That's definitely not easy. And that's complicated. So, spatial tissue profiling might seem very, very new, and this is our mindset. Everybody has their own. But and it's a methods of the year 2020 for spatial transcriptomics, 2024 for spatial proteomics. But it might seem exciting and new, but it's practically, a part of a series of innovations. People have been investigating tissue for thousands of years, from the time of Aristotle and before, with naked eye, with the magnifying lens, with the microscope, and they're like dozens of paradigm shifts that changed the world at that specific point in time. And then they gradually, in the 1940s, IHC and IF, they seemed space age, but now we do, millions and millions of slide stain per year across the US. And so they're this technological breakthrough, and then they reach, research and translation. So, and of course, clinical application. But now it's a different world. We have a series of paradigm shifts, and they build upon, the explosion in capabilities, microscopy, sequencing, AI, ML, data analysis in general. So now it's a series of paradigm shifts in a very short amount of time, which makes it very, very exciting. So spatial tissue profiling, we see it as part of that, continuum. And because it's a new technology, it majorly started, through early discovery, but we see them now, moving very quickly to translation. For clinical, that needs, more work to be done. And what are the major obstacles, for both research and translation? And that's a wall, it's a new field, and that's a wall we have to climb together. with many aspects and many milestones to be met. We don't have standards as a field yet. There are like ways to validate turnaround time. Some experiments, especially for clinical trials, if you want, or um, you know, it can take a while, and that's it's for clinical application. That's, for instance, absolutely I know, no, and we want to be able to support easily FFPE, lower quality samples, to be able to automate, much better than now. High throughput, a clear, pathway for easy adoption in different spaces, in high tech spaces, in community hospitals, in small and bigger research labs. Um, you know, how to train people more easily, and the cost per slide is a major deterrent for translational, for research, for um, so from this, point of view, when we assessed the G4X, when we first were with the opportunity to bring it in, we thought that, its speed, FFP centric approach, very high throughput, at least on paper. Back then, we haven't seen the instrument working, and the, promised very low cost per slide. I think they check some of these major boxes, which I think are part of our optimization process to make this field bigger and more relevant to every aspect of research, from early discovery to translation to also clinical application. So back in like December 2024, when we brought it in, we joined forces with the lab because the lab had some interesting questions to try on the instrument while we're SOPing, and we put multiple instruments in, and people from the lab and the facility. So that was a relatively large investment on our end to see what we can see. So there is a lot of intensity in trying forward. So there are three instruments currently in the facility. Those two, they live side by side, and the other is actually in the corner, like here if you could look, but you cannot. And we try to give all of them prudential view, which everybody knows, it increases data quality. The prep now, our experience, and I will go like from very, very practical things, and I will try to use questions I have received from colleagues, ABRF, Gestalt. I will discuss about them in a bit, but also online. The prep is definitely very different, and it's quite interesting. Um, so that's all my personal view. So there is this step where you transfer tissue using hydrogel punching, which seems very intuitive, like, oh yeah, for sure, but actually it's very novel. I don't know why as a community we weren't doing this before, but I think it's very, very convenient, especially if you're trying to use a lot of samples. The main drawback for this, and again, that's a personal opinion, is that, every time you're using a proprietary slide format, every time you're using something that's not the standard pathology slide, um, that, complicates a little bit. People would want you to cut. But for instance, you want the block, but the block cannot leave, the biorepository where it's in. So that is resolved because the gel pads can last for a month. So they can load in the gel pad. We get the gel pad from our collaborators or clients, and then we can like get the bunches from there. Um, but that was something, that we needed to see in action how it works, and that's a good workaround. But that, for instance, is just something to take into account. The second thing, and that's not as intuitive as the first one, is that the throughput is very high, and it seems like a good thing, but you have actually to rework. We had to rework our facility to be able to, for instance, when you have this small size, so this is a small size format. These are 4.5 by 4.5, and you're putting 32 of those per flow cell, and the instrument has multiple flow cells. So if you're doing an experiment with 64, sections, and if you're doing the large format, this is 10 per flow cell, and you have four flow cells. So if you're doing an experiment with 20 or 60 or 64 sections, this means you have 64 blocks to work on. You have to section 64 blocks. You have to float, near water bath, 64 sections. So we have currently a football field equivalent of water baths, for instance, and plenty of bench space. Um, of course, that's asynchronous. So you can do this asynchronously. So and we do this asynchronously. You have people loading the flow cells and prepping the samples, and then you have also people like working the actual experimental workflow. And you can, load and store. It doesn't have to happen, concurrently, as I mentioned, this day a month. But that is something, that if you haven't thought about this, that's one of the first things that you will need to think. The other part that needs to be thinking a little bit about is how can I be using the real estate every time? So if you have 32 and 32, for instance, if you're starting with 64 samples, and this is like when you're loading them on the instrument, or if you're, um, or if you're doing, five and five, but um, you will not always have a project that has 64 or 32 samples every single time. The good thing is that, for instance, in this four-lane format, let me go back. In this four-lane format, every lane can have a different probe set. So you can have breast, colon, lung, something else, um, become very different. Uh, but that needs some coordination of your projects. You have to have all the blocks at the same time. You have, every time to know that you have. So I think it's a similar when you have a sequencing core facility and the sequencers become bigger and bigger. Now, but it's easier, for instance, to pilot. For instance, if somebody wants to try, they can get only one lane. Or if you want to test, like, are the samples good? You don't have to go full flow cell, especially if you can put those together. So how many samples have we run? Uh, and since December, hundreds and hundreds of samples. Since March, when we finished testing and SOPing, and the I will discuss a bit, and the chemistry started becoming more of final phase, we started offering it as a service in the facility. So many of the samples we did were also for client projects and for consortia and for other things. What I can definitely disclose is that even though the chemistry has been updated multiple times and the instrument has been updated multiple times since then, it's pre-market. It's early access. If people ask me about, is it robust? So we haven't lost a sample, and we haven't, through detachment, for instance, and we haven't lost a run. And why is this important? Because the bigger the sequencer, similarly, the bigger the spatial runs you do. If you lose 64 samples, for instance, that's a lot of samples to lose as a facility and as a PI as well. So, like, knock on wood, until now, we have run hundreds and hundreds of samples. We haven't, lost a run. Um, yeah, which I think is an interesting statistic. Um, and well, let me see what we have here on this slide. So we have liver. In this one, we have liver. We have breast cancer biopsies, and we have marrow on the bottom. Let's see what else we have here. So this is another flow cell, same run. Again, we have liver. We have marrow, and we have skin. So this is how we usually also, put things together, just because you're trying to get the maximum real estate. We get a lot of questions about marrow. So bone marrow, we have tried formic acid, EDTA, nitric acid. We've been skeptical about hydrochloric acid. We might give it a go again. Um, we had bad experiences with other assays, um, but for everything that I mentioned, we've had good results out of the box, no optimization. And these are decalcified human, archival. And this is around the Maro sample that I'm just showing with standard. That's not EDTA. That I think is nitric acid. Um, so the other thing I mentioned, versions multiple times. I don't know if Singular wanted me to show this or not, but this is the reality of our experience with it. So there are three major version changes that we experienced December, um, January, and February. So the assay kept, like, changing, and you can see, for instance, especially for transcripts per cell. I mean, the cells did not change, but for transcripts per cell and genes per cell, every version increased a lot. So again, of course, this is pre-market, and it's not, how the whatever is going to be out and whatever you will be working on will be. But we saw that, within 3 months, we saw like more than double the transcripts. After that, now there are some improvements since then, but I think it's more like a small percentage of improvement further than that. These are the three versions, and you can see the difference that I mentioned previously. What was very interesting for us to see, and I think, Singular shows plots like this, but when we have the assay in our hands, we do it ourselves. Correlation, zero sections. These are zero sections, uh, same block, same run, and we see good correlation in the target probes, but also in the negative probes. And this is an interesting one, a little bit more complicated to explore, but this one is runs like different runs, serial sections, maybe a couple of sections apart, but these are different versions of the assay. So we wanted to see when we saw that improvement, in sensitivity, do we see also different things? No, we just saw more. Yeah, so that was, for instance, what we noticed. Um, and I will now switch gears and switch hats and discuss a little bit about um, how we use the assay for the lab. So we have multiple projects at the moment in the lab, and we use G4X, and many are clinical trials of the cancer research efforts, the standard projects that we do. Those that fit, usually those that have large, for instance, populations that we want to test, or those where we want to very like a low cost pilot a set of samples, see if there is signal. So I will be explaining that. I think here is a project that checks a lot of these boxes. That's why I chose this. I chose a single project to discuss, but I think you'll understand why. I don't know if you're familiar with HuBMAP, the Human Biomolecular Atlas project. So our lab is part of an NIH HuBMAP Tissue Mapping Center. HuBMAP is a fast scale effort. You can check their website; is awesome to redefine human anatomy using spatial tissue profiling and single cell technologies. And our tissue mapping center started focusing almost exclusively on the lymphatic vasculature, but we expanded during the past couple of years to all vessels across the human body. And this like tissue mapping center brings together experts from the Boston Lymphatic Center, Dr. Drew and Dr. Tsai, from the Center of Excellence from the UPenn, Dr. Itkin, Dr. Young Hong from USC, and Young moved to BI actually quite recently. Welcome, Young, and the Spatial Technology Unit and colleagues like Gordon Chang from our uh, medical center. So and all of us we joined forces to perform this like multi-tissue endothelial cell atlas. Now we, as I said, we work lymphatics and vasculature, and most tissue mapping centers in HuBMAP are focused on the single organ. That's not the case for us. The difference for us is that we focus across a large number of different organs, and that puts a lot of pressure for people who are doing spatial. That's not the easiest thing to do to have like different organs, different tissues, different panels, different genes of interest. And of course, it also increases the cost. The more tissues you have to optimize for and work with, that increases the cost. Um, so we thought that G4X could pick up some of that burden because also for HuBMAP, everything we have to do is in 3D. So or either natively 3D, as here you can see these two are light sheet, or serial section 3D. Above it is codex, and we do serial sections, and then we register and make 3D objects. Spatial transcriptomics is especially good for a project like this because you can switch the panels very easily. You design them with the team from the single cell data you have, and then boom, you're off. So I picked one of the organs that we work on, and that's heart. And this is a collaboration with the Heart Tissue Mapping Center, with doctors Liming Pei, Kai Tan, and Ken Margulis, who also provided the samples. This is a beautiful heart sample that they gave us. And this is another section. This is the left ventricle. And we designed a custom heart panel based on our tissue mapping center needs and programmatic obligations. And now this is very early data. The cluster calling you see and what the annotation was done by Patrick in the lab. And what he did was he label transfer from the human cell atlas heart directly, no optimization, nothing, so that I can have some nice slides to show you today on these hearts. And it actually works really well. So there's good correlation between single cell and the spatial profiles. So blue is heart muscle, orange is epicardial fat. You have some nice vessels, here and there with different colors. Purple is vein, light brown, is artery. The darker brown is smooth muscle. Um, it looks, really nice. I have some zoomed in, some nice like complex architectures that we work on. I mean, we work with vessels, so I will be showing you vessels. That's what we like a lot. So here you see this artery with light brown, like monolayer it's actually an arterial, a monolayer of endothelial cells around it, a monolayer of smooth muscle. You have this like very nice lymphatic vessel here. Green is fibroblasts. A vein again, monolaries here have some macrophages with yellow, and blue is your muscle. That's another section. These are we're using the 1 centimeter, so that's a 1 square centimeter, and you can do up to 10 per flow cell, multiple flow cells. So these are the 1 square centimeter, and this is you see this like massive vessel over here, and of course we zoomed in because that's what we care about. And you have like this artery, a lot of muscle around it, fibroblasts. You have a vein right next to it. This nerve bundle over here with pink. You have some lymphatics here and there, just a couple of cells, which you would have missed with just H&E. Another artery here. I think is yeah, if that's like automated annotation without us doing anything in cluster mapping, that's a good start. And of course, why we chose the G4X for this is the ability to very easily do a lot of sections from the same organ to do 3D, because that's a programmatic obligation for HuBMAP. So we're doing multiple sections between us. It's also one of the easiest ways to use the G4X real estate, because if you have one block and you do 4 or 5 sections, 10 sections, you want to do 3D, you don't have to prepare another block, face a block, start cutting the other block. Of course, it's a few minutes difference, but I think the team enjoys a lot in serial sectioning and loading. Um, I think it's a bit more convenient. So that also, if you use less labor, less hours, we're a cost recovery center. We work at 0% profit, and so everything has to be exactly costed out. So if you spend less hours working, it actually reduces the cost, and that's what the lab likes as a client. Um, so yeah, so we're now in the process of creating 3D objects. People ask us, what are you using for alignment? We're using STLine. That's fun, and we're using also Voltron, High-Throughput. Um, it's a really cool tool. Give it a go. So another good option, I think, is let me see. We're using it for tumor work. We have members in the group that are using it. We have a lot of tumors, for instance, very large cohorts, especially for rare tumors. The team spends a lot of time collecting rare tumors for the research questions we have. And that's, a good option to just run all of them, not just 5 and 10. Um, we have projects as a lab. I cannot discuss projects in the facility because there are other people's. We have projects in the lab where we have high tumor heterogeneity, or we have multiple time points. We're following them longitudinally. So these are, good matches to have the ability to do a lot of samples in the same project. So what's next? I told you I'll be very transparent of what we're doing now, what we did up to now, and what we're doing in a bit. So, and that's the first time I actually sharing what our plans are with such a detail. So the RNA and protein assay, we're super interested in this, but we opted out from sending samples to Singular, which you can. If you want, we wanted to see the assay on the bench, and we're going to do this next week. So we have our training September 22nd week, and we have I think a solid plan to. This is from the vendor, from the website. That's where I stole it from. That's not ours. So we have a plan, and feel free to reach out and ask. But also, I will either through ABRF or Gestalt or bioRxiv will provide all this information. So we'll be testing RNA and protein correlation. Of course, RNA and protein do not correlate very well. You have post-transcriptional regulation event. You have a lot of things happening, you know, for the RNA to become protein. But that's one thing that we will be testing. How protein helps segmentation. I think that's a very interesting thing to see. Um, We will be doing some chromogenic assays and some multiplex IF to see, if I have an assay that's only focused on protein and I do serial section, what's my concordance? And of course, everything will also pass from our team, but also through domain expert evaluation to know exactly right. So I think we that's also I think useful for you regardless of the instrument if you're interested in the instrument. So we will be using to validate the what we see with the antibody capabilities of the instrument, what we also did and still do for our Akoya, for instance, pipeline, our codex work, um, everything we do for protein work, we follow this manuscript. This was led by wonderful people from HuBMAP. We're happy to be collaborators in this and participate in that massive effort. Um, there is a portion in this manuscript that captures AVRs, how to do antibody validation reports, how to validate multiplex antibody panels, how to know that this is a good antibody panel that works really well, what type of reporting you should be doing, and what kind of internal controlling you should be doing. So when we do this on the September 22nd week, we will be evaluating with the same standards that we use for production for assays that we have a lot of experience with, and we'll be following, what we co-wrote in this paper, what we think is a good idea to be testing. Direct seq now, that I cannot disclose much since we haven't tried it yet, and we're starting now. We will be focusing on the TCR, BCR capabilities of the instrument, on FFP, and that's super cool. But we haven't tested it. Tentatively excited, as with every new technology. But when we see it, happy, reach out, ask us. We're always, providing, happy to provide feedback. So what I'm sharing now, and they're like, you see there like watermarks there, even though it's not relevant today, it's a blast from the past. And this is not, an official activity endorsed by Singular, and I will explain why. Um, we are doing now official benchmarking of the instrument. Why we're doing it now, the official benchmarking? Because the chemistry is now finalized. And we thought it didn't make any sense you know, to benchmark something that is work in progress. But at the same time, when we thought that, our SOPs are mature, and I think we thought, the community would have interest in this, we did our internal testing as the test that I showed you previously with serial sections on the same run and between different runs, um, to see how the instrument behaves and whether it's worth actually bringing it to the community. So we did internal testing, and that's the results of the test, and that's why I have the watermark so it doesn't make it to social media as this is the behavior of the instrument today. This is the second generation chemistry. We're now 3 plus. And we compared it against xenium that we have in the lab. These are serial sections, breast cancer samples, and you know, again, the benchmarking that we're doing now is much detailed and large scale effort. And in a couple of months, we will provide very accurate information through Gestalt, most probably. However, happy to share this information when we had it. We did this testing internally for us, um, and and it seems like very good concordance between the two assays. Like for instance, this section is like, even same scale, same everything. We expect the results to be a little bit different now in terms of how many transcripts per cell on the G4X because we have seen this. But apart from that, we were very happy to see. We have a lot of faith in Xenium. We have done hundreds, of samples. It's an instrument we use for a very long time, and we're very, familiar with it. So we were happy to see that, this instrument we were just starting back then, and we did the first few runs. We managed to get a good quality of results from it. So I mentioned the BRF Gestalt multiple times. These are acronyms that actually mean something. If you're running a core facility, you will probably all know ABRF, which is the relevant international association of core facilities, and it's a phenomenal resource and has amazing WhatsApp, platforms and people that discuss core all day. Gestalt is a relatively new, so I'm happy to make a brief shout out and intro. It's a global alliance for spatial technologies, and the goal is just to bring, people from different walks of life that are interested in spatial. So Gestalt was established 2 years ago, so it's very new, and currently brings together more than 1,500 people from 40 countries. So it grew very, very rapidly. It has a good representation of mix of labs, cores, industry. Um, Personally, my lab members, as well as the core facility members, benefit a lot from the information exchange that takes place. There are a lot of working groups that are currently being, starting. So if you're interested in spatial and you see yourself as part of a community or you want to be one, it's free, it's fun, and we have a wonderful seminar series. It's one seminar per month, and if we have some results from the benchmarking, maybe we'll find some time there, or we might use the alliance some way to make them faster than bioarchive. Of course, there will be a bioarchive, um, but if people want to see the results faster from the benchmarking, happy to use the some space or some time from the alliance to make that happen faster. So check it out. Again, a cool talk happening end of the month for Gestalt, and tune in. So at this point, I wanted to thank, of course, the lab members. With Purple are the lab members who are already using like routinely G4X data for their work. With Blue are the folks that they have decided that they want to use it for um, for something regarding their work. They're designing a panel at the moment, or they're getting the samples to work on this. I had a shout out to Patrick because he's from the lab and he was responsible in optimizing the spatial transcriptomics capabilities of the lab to seamlessly work with G4X data. Of course, I wanted to thank the Spatial Technologies Unit, which is where everything happened, the training, the rollout of the instruments. Shosha and Antonella performed the experiments. And Sheetal, who is the computational biologist tasked to roll out the instrument, there was a lot of work involved in that. I wanted, of course, to mention, the HuBMAP team, um, you know, our collaborators, our funders, of course, the patients and their families. These are, human samples we work on majorly, and it's the most difficult times of their lives, and they think about, supporting others through the donation of their samples. The lab is super collaborative. If you want to reach out, you can find my information there. And if you're interested in the facility, you can reach out to them through their website. They're awesome. They will help you. We're looking forward to more results for the platform. And I'm almost like 30 seconds before the 45 minute mark. So we're on time. Thank you for staying hopefully till the end. And if you have any questions, you can easily find me online on Gestalt, on WhatsApp, LinkedIn, X. Happy to share any other opinions. Take care. Have a great day, everyone.